Hello everyone and welcome to our latest Biomarkers webinar. My name is Jessica and on behalf of Oxford Global, I would like to thank you all for joining us today. Today's webinar is on the subject of non-invasive clinical biomarker discovery and development with the breath biopsy OMNI assay. Our first presenter is Jeff Buckthal, Senior Business Development Manager at Owlstone Medical, who will present on Owlstone Medical and the breath biopsy platform. This will be followed by Mohney, Head of Clinical and Translational Science at Owlstone, who will present on wide-ranging clinical breath biopsy applications for the pharmaceutical industry and academia. After the presentation, we will have a 10-minute Q&A session. Please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. Without any more delays, I would now like to get started with the presentations. Jeff, over to you.
Thank you all for joining our webinar, Non-Invasive Clinical Biomarker Discovery and Development with the Breath Biopsy OMNI Assay. Our goal is to give you a better understanding of breath as a rich new source of biomarkers that can be reliably detected and measured using our breath biopsy system. Before we jump into that, some quick background on who we are. Owlstone Medical's mission is to save 100,000 lives and $1.5 billion in healthcare costs. And the way we plan to do that is by using breath biopsy to develop diagnostic tests for early detection and precision medicine. We have our own programs currently focused in respiratory diseases, liver disease, lung cancer, and digestive health. And we also collaborate with pharma, biotech, industrial, and academic partners across a wide range of disease areas to address their specific research or clinical questions. Here are some examples of the disease areas we have worked in, along with our collaboration partners. And on the right are some illustrative results broken down by application, either early detection or precision medicine, along with the patient numbers and performance. So what exactly are we measuring in exhaled breath? We are measuring the volatile portion, and more specifically, volatile organic compounds, or VOCs. These can come from two sources: endogenous, where they are the end products of metabolic processes and are affected by both physiological and pathological mechanisms, or exogenous, where they are introduced through external factors such as diet, drugs, exercise, smoking, vaping, and the microbiome. A breath sample, therefore, represents a snapshot in time of a patient's phenotype and can be used to non-invasively monitor patient groups as they progress through various exposures, diseases, or treatments. Breath analysis doesn't just report on the lungs, it offers the opportunity to collect a sample that is representative of tissues throughout the whole body. VOCs can come from not only local cellular metabolism, lung tissue, and the lung microbiome, but also systemically, as VOCs exchange readily from circulation into the lung alveoli and can be measured on exhaled breath. This slide really highlights the utility of breath to provide useful biomarkers outside of just respiratory research. If we look at the cumulative number of clinical trials involving breath analysis, there are a broad range of therapeutic areas covered. Why breath? We think there are quite a few compelling reasons to consider breath analysis. It's completely non-invasive, pain-free, and well-tolerated. Unlike with tissues and biofluids, there is an inexhaustible supply. VOCs represent what is actually happening phenotypically, providing a dynamic view of current metabolism and disease biology. Our process that I will describe later allows us to identify the VOCs present rather than relying on pattern recognition, which can be difficult to relate back to underlying biology. By identifying the VOCs, we can relate these to biological processes. Our process allows for pre-concentration, so detection limits are in the parts per trillion range. This is particularly important in early detection where potential biomarkers are likely to be low abundance. Since it's non-invasive, high-frequency serial sampling is possible with no ethical or patient comfort concerns. As I mentioned earlier, breath is for more than just respiratory disease. Breath could also be used as an orthogonal approach to other biomarker platforms. And lastly, because breath is so easily accessible, there is the potential to translate to point-of-care or even home-based testing in the future. Where are we currently in breathomics? Modern breath testing started with Linus Pauling back in 1971, and over the last 50 years, there have been hundreds of scientific papers suggesting the presence of VOC biomarkers across a range of diseases. One thing that becomes clear in going through these papers is that there is little agreement in biomarkers associated with particular diseases. That probably answers the puzzling question of why breath testing is not yet used routinely in clinical settings. Another thing that becomes clear is that there was no standardization of breath collection and analysis. These homebrew methods really lack the hardware and protocols necessary for robust, repeatable breath sampling that is necessary for confident biomarker discovery and validation. This challenge is what we have addressed with our breath biopsy system, which allows for the first time the reproducible collection and analysis of breath samples. The collection side consists of the Receive a breath sampler, the Casper portable air supply, and the breath biopsy collect software. The analysis side consists of a high resolution, accurate mass, gas chromatography, mass spectrometry system. I will discuss each of these components in a bit more detail over the next few slides. Reciva is the first standardized device that enables patient breath samples to be collected reliably and reproducibly. During sampling, patients just breathe normally while Reciva tracks their breathing pattern. This is done with pressure and CO2 sensors that you can see in the schematic. At the appropriate breath fraction, pumps are activated and pull the exhaled breath onto the sorbent tubes. Over a period of time, Reciva traps and stabilizes the VOCs and pre-concentrates them onto these tubes, which are a bit like a vacutainer for breath. These are then sent to our breath biopsy lab for analysis. Reciva has been deployed in studies at over 100 clinical sites and hospitals around the world, including the world's largest breath-based clinical trial. Here's what a breath collection looks like. The Reciva has CO2 and pressure sensors that monitor tidal breathing in real time, shown here at the top. The Reciva also has two pumps, so what you see in the middle is that during expiration, the pumps are being activated sequentially. So in a single breath collection, there are two different breath fractions captured, alveolar and bronchial. The pumps are programmable, so it is possible to collect the whole breath sample. Reciva collects a set volume of breath, which takes approximately 10 minutes. Since our blood volume circulates through our lungs about once per minute, there's quite a large headspace of blood collected in a given sample, and this pre-concentration enables much higher sensitivity. Another component on the collection side is the Casper portable air supply. A real challenge in breath analysis is the inclusion of environmental VOCs in breath samples. These can come from things like air pollution or cleaning products and can be at much higher levels than endogenous VOCs, so they can interfere if included in a breath sample. Environmental VOCs also vary over time and across clinical sites, making inter-site comparisons difficult. And consider that the room where the breath sample is collected will contain VOCs from any other people breathing in the room. It won't be possible to know if a VOC is a true on-breath compound or something coming from the environment. Until we developed our breath biopsy system, breath samples were typically captured in bags. Patients would inhale air from the room and then blow into the bag. These samples contain a lot that isn't really representative of a patient's phenotype, and this is probably why many previous breath studies have not been reproduced in terms of the biomarkers identified. Casper filters the air patients are breathing, which significantly reduces contamination by environmental VOCs. The scrubbed air is supplied by a tube that goes from Casper to Reciva. The graph on the bottom left shows a comparison between a sample collected with and without Casper. With Casper, there is a significant reduction in the overall signal coming from the environment. In the middle graph, you can see that with a clean background from Casper, the sensitivity is much higher than with a contaminated background, where there is little difference between the VOC level between what is on breath or from the environment. On the right, you can see how Casper reduces false discovery, as this VOC is clearly not on breath. It is coming from the environment. So the combination of Reciva breath sampler and Casper portable air supply allows for the reproducible and reliable collection of breath samples. The final component of our breath biopsy system is a standardized analysis platform we have developed, which includes the gold standard for biomarker discovery. thermal desorption, gas chromatography, mass spectrometry. We use a high-resolution, accurate mass spectrometer that has been optimized to enable high-quality VOC detection. The high resolution maximizes the potential for detecting a wide range of VOCs covering many chemical classes, and the sensitivity allows for detection of low abundance biomarkers. Although HRAM, or high resolution accurate mass gas chromatography mass spectrometry, is a gold standard for biomarker detection, it does require standardized protocols to ensure reproducibility and confidence in the data generated. We've developed our OMNI assay with this in mind. A key feature of this is our breath biopsy HRAM library, which consists of VOC standards that we have run across our platform. Since we know the signature of each VOC standard, we can match it to the VOC signal in a breath sample and make a high confidence call on each VOC identity. For many of these VOCs, we may know the biological origin or function, which allows us to build targeted panels in specific disease areas. Here's an example of a targeted panel. This is our respiratory panel, and it contains VOCs previously implicated in chronic inflammatory diseases of the lung. So what does a breath biopsy study look like? We will provide a collection station at each clinical site, along with online training and support throughout the trial. We will also provide a regular supply of breath biopsy kits. Each kit contains a single-use mask and the breath biopsy cartridge for collecting a breath sample. Samples and subject data get collected in your clinic. The subject metadata then gets securely transferred to our breath biopsy digital biobank, and the cartridges get shipped to our lab at ambient temperature for analysis. Shipping on dry ice is not required. We will then analyze the samples, and depending on the project needs, we can provide comprehensive statistical analysis and biological interpretation. Final slide from me. We will be holding our 4th annual Breath Biopsy Conference this year on October 12th and 13th. The purpose of this conference is to facilitate progress in the breath research field by bringing together leading researchers and research organizations to share the latest advances and discuss common challenges. The key areas of expertise in and around the breath research field will be explored in talks, poster sessions, and discussions. The conference will be virtual and is free to attend, so we look forward to your participation. Registration and abstract submissions are open now. And now I'll turn it over to Rob Mohney, who is Owlstone's Head of Clinical and Translational Science.
Thank you to everyone who's joined us for this webinar. Before I present results from our non-invasive biomarker discovery program, I'd like to reiterate a few points. As previously mentioned, breath is a rich and complex mixture of volatile and non-volatile components. The volatile component of breath contains VOCs that are produced by metabolism in the body and transferred through the blood to the lungs, where alveolar gas exchange allows for their release in the exhaled breath. Non-invasive profiling of breath VOCs can provide a source of useful biomarkers with clear association to the body's metabolism. By comparison, the non-volatile fraction of breath consists of respiratory droplets and aerosols that contain a variety of biomolecules. In the interest of time, I won't present the non-volatile fraction of breath in any detail today. However, additional information on its use for the detection of disease can be found on the Owlstone Medical website. The origins of volatile metabolites on breath are diverse and can reflect a variety of factors. VOCs may originate from endogenous sources where they reflect metabolic and disease processes in the body, some of which are influenced by our genotype. VOCs also originate from exogenous sources, molecules that are ingested through our diet or from environmental exposure, medications we may be taking, and even the microbiome from our gut and airways. These exogenous molecules may provide very reliable signals about processes in the body because our bodies metabolize and detoxify these molecules to clear them from our system. With the technology we've developed, we're able to capture on-breath VOCs to get a snapshot of the underlying phenotype and to identify metabolites that are reflective of aberrant metabolic activity, which could represent biomarkers for disease. It is our mission at Owlstone Medical to identify and quantify these markers and convert them into useful clinical tests. Here's one example how VOCs may provide a readout of the underlying phenotype. Factors such as aging and smoking, and even conditions such as hypoxia and inflammation can lead to a disturbance in the balance between our antioxidant defenses and the production of reactive oxygen species, resulting in oxidative stress. Indeed, as we'll hear today, this is a common feature of many diseases, some of which are listed in the table to the right. When present in excess, reactive oxygen species can react with and degrade membrane lipids through the process of lipid peroxidation. Some of the products of lipid peroxidation are very reactive themselves and drive further biological effects. Many of these products, and indeed even some of the drivers of lipid peroxidation, are volatile molecules that can be measured on breath, such as the aldehydes. The precise VOCs produced can be disease-specific and form the basis of a VOC panel for disease detection or treatment monitoring. Today, I'll present a few case studies that highlight the applications of breath VOCs as biomarkers for early disease detection and precision medicine. I'll start by detailing some of our own work to validate a biomarker for liver disease and to develop a clinical test for detection and monitoring of liver disease progression. Next, we'll explore the role of VOCs in precision medicine with a focus on differentiating types of chronic inflammatory airway disease to better direct appropriate treatments to patients. And finally, we'll look at how VOCs could be applied to pre-screen former smokers and help differentiate those with and without lung cancer. Let's start with liver disease. There are many types of liver disease. Fatty liver is one type characterized by the excessive accumulation of lipids in the liver. This condition can be caused by alcohol consumption, though this phenotype isn't our focus today. Fatty liver can also be caused by other factors, such as obesity and elevated blood glucose or triglycerides, leading to non-alcoholic fatty liver disease or NAFLD. Some individuals with fatty liver may develop a more severe form called non-alcoholic steatohepatitis or NASH, which is characterized by increased hepatic inflammation, ballooning, and fibrosis. Most people with NAFLD or NASH live with this condition for many years without experiencing symptoms. And up until this point, changes in diet and lifestyle can help manage and improve the underlying condition. However, undiagnosed disease can progress to more severe and irreversible conditions, including cirrhosis, liver failure, and liver cancer, resulting in premature death or requiring a life-saving liver transplant. Improved early detection of liver disease could save lives by revealing this condition at a time when preventative action can be beneficial. The current gold standard for diagnosing liver disease is a biopsy, which is invasive, expensive, and comes with an increased risk to the patient's life, which makes biopsy impractical for the use in early stage liver disease detection. There are several reasons why on-breath VOCs are better suited to detect and monitor liver disease, but they could be applied more routinely to otherwise healthy individuals in a primary care setting. For starters, the liver is metabolically very active and central to many of the key metabolic processes in the body. Among other functions, it is responsible for the storage and release of key nutrients, detoxification of harmful substances, and the breakdown of red blood cells. The liver also receives a rich supply of oxygenated blood, as well as a secondary supply of nutrient-rich blood from the gut. This combination of rich blood supply through the liver and high metabolic activity provides ample opportunity for volatile metabolites to enter the bloodstream and get carried to the lungs where they can be released in the exhaled breath. Given its vital role in central metabolism, damage to the liver is expected to alter the levels of metabolites in systemic circulation and on breath. And indeed, researchers have shown that specific patterns of breath VOCs have been associated with various liver diseases. These VOCs reflect underlying disease mechanisms like oxidative stress and lipid peroxidation, as well as changes to the cytochrome P450 system that is responsible for oxidation of endogenous and xenobiotic compounds. A review of the literature has found a handful of studies that compared the breath VOC profiles of cirrhotic patients with that of healthy volunteers. In general, these studies reported mostly non-overlapping biomarkers on breath, with the exception of the volatile compound limonene, which was elevated in cirrhotic patients. This caught our attention because limonene is a molecule of dietary origin, typically found in citrus fruits and many vegetables. It is used as a flavor additive in the food and beverage industry and as a citrus fragrance in cleaning products and air fresheners. There is no evidence that limonene can be produced in the human body. However, exogenous limonene is metabolized and cleared from the body through the activity of cytochrome P450 enzymes, the levels of which are reduced in patients with cirrhosis. What is the evidence supporting limonene as a biomarker for liver disease? A couple of studies from Professor Chris Mayhew's lab at the University of Birmingham provide the most compelling evidence as shown here. On the left, we see that levels of limonene on breath are significantly increased in patients with cirrhosis compared to healthy controls. Interestingly, as shown on the right, levels of limonene on breath of cirrhotic patients were observed to normalize over time after liver transplantation. These data suggest that limonene functions not only as a biomarker for liver disease, but also for liver function. Moreover, if correct, limonene may provide a readout for monitoring the effectiveness of therapeutic intervention for liver disease. We wanted to independently validate the results from the literature. To do so, we ran a pilot study utilizing an independent population drawn from Owlstone Medical's pan-cancer study. This is a prospective cross-sectional case control study involving a range of cancer types, including liver cancer. Using TD-GCMS, limonene was targeted for identification and quantified by comparison to a limonene standard curve. The results from this pilot study were published this past fall, and I'll cover a few of the highlights from this paper today. First, we measured breath limonene collected from control subjects with no known liver disease and from patients with cirrhosis, both with and without hepatocellular carcinoma, or HCC. In the box plot, we see that patients with cirrhosis had significantly elevated levels of breath limonene compared to control, confirming data published by other researchers. We next wanted to know whether the presence of liver cancer has an influence on breath limonene levels in patients with cirrhosis. For this, we separated cirrhotic patients by liver cancer status. That is, those patients with only cirrhosis versus patients with cirrhosis complicated by hepatocellular carcinoma. It's important to note that the two disease groups were matched for several parameters, including age, gender, and the severity of liver cirrhosis. In the box plot, we see that the middle group of patients with cirrhosis had significantly elevated levels of breath limonene compared to controls. This was also true for cirrhotic patients with hepatocellular carcinoma, as shown on the right. However, if we compare the two liver disease groups to one another, we find no significant difference in breath limonene levels, indicating that liver cancer does not influence limonene levels in patients with cirrhosis. Given this result, we pulled the cirrhotic patient groups back into a single group for further analysis. We wanted to estimate the performance of a potential limonene breath test to identify liver cirrhosis. For this, we calculated a receiver operating characteristic curve using breath limonene levels from control individuals without liver disease and the combined group of patients with cirrhosis and cirrhosis complicated by HCC. The resulting analysis yielded an area under the curve of 0.78 and a sensitivity and specificity of 73% and 78% respectively. We next investigated whether levels of breath limonene were correlated with clinical biomarkers of liver dysfunction in blood. For this, we examined 4 clinical markers, the first 3 of which provide a readout of liver function, and the last of which provides a readout of liver damage. The first is bilirubin, which is a catabolite of heme derived from the breakdown of red blood cells. An increased level of bilirubin in the blood is a marker of reduced liver clearance. Next is albumin, the most abundant protein produced by the liver. Low levels in the blood are indicative of chronic renal failure. Also, we have INR, or International Normalized Ratio. This is a normalized measurement of the time it takes to convert prothrombin into active thrombin during blood clotting. An increase in INR reflects lower levels of prothrombin synthesized by the liver. And lastly, we have ALT, or alanine aminotransferase, an enzyme found mostly in the liver. When liver cells are injured, ALT is released into the blood. It's important to note that while these 4 are used as markers of liver disease, they are nonspecific and can be altered by conditions other than liver disease and liver injury. So what did we find? Using a regression analysis to correlate breath limonene levels with blood metrics, we found that limonene was positively correlated with bilirubin and with INR, and negatively correlated with albumin. No correlation was observed between ALT and breath limiting. These data suggest that elevated limiting levels in breath behaves as a marker of liver function rather than liver damage, thereby addressing an unmet need for non-invasive markers in this area. Bilirubin, albumin, and INR are used in conjunction with other clinical markers to calculate a Child-Pugh score and assign a Child-Pugh class, which is reflective of the severity of disease in patients with cirrhosis. Individuals classified in Group A have less severe disease than individuals classified in Group B or C. Those in Group C have the most severe disease and the low survival rates. When we stratified cirrhotic patients by Child-Pugh class, we found that breath limiting levels in Class B were significantly greater than those in Class A, consistent with the increased severity of disease in Class B. So thus far, we've demonstrated limiting as a non-invasive biomarker for liver cirrhosis and shown that limiting can stratify patients by disease severity. We've also shown that levels of breath limiting correlate with existing clinical markers of liver function rather than liver damage. So where do we go from here? Up to this point, our investigation is focused on late-stage liver cirrhosis. We now need to extend these findings to earlier stages of liver disease, such as NAFLD and NASH, where there's greater opportunity for therapeutic intervention. One strategy we're employing to increase our capability to detect early disease is to develop limiting into an exogenous volatile organic compound probe, which we refer to as an EVOC probe. EVOC probes are substrates that aren't produced in the body. However, when they are ingested, they can undergo metabolism and be excreted on breath, where they are detected with high sensitivity. An EVOC probe is selected to assess the metabolic activity of a specific enzyme in vivo based on known mechanisms of disease. For example, chronic liver disease is known to downregulate the cytochrome P450 system, including CYP2C9 and 2C19, which are reduced in patients with cirrhosis. Limiting is specifically metabolized by these two enzymes, thereby providing a direct readout of the aberrant metabolic state associated with liver disease. The EVOC approach can offer some compelling advantages. For starters, it is completely non-invasive and works with existing breath biomarker kits. Next, the lack of endogenous sources of these VOCs results in a clear signal and higher sensitivity than detection of VOCs with endogenous origins. In addition, multiplexing is possible. Because each probe is targeted for conversion by a specific enzyme, one can theoretically administer a cocktail of EVOC probes to assess multiple enzyme targets or test for multiple disease states. And finally, many of these substrates are readily available compounds, which should make them more cost-effective and ease licensing requirements. In addition to the targeted EVOC probe strategy, we're also applying the non-targeted OMNI assay to identify additional VOC markers for liver disease. Here, our goal is to augment limiting with additional markers of liver disease that will allow us to achieve high clinical performance in terms of sensitivity and specificity. So we applied a non-targeted method to profile VOCs in breath using the same samples derived from cirrhotic patients and controls taken from Owlstone's pan-cancer study. The non-targeted approach identified 270 additional VOCs, of which 18 were significantly different with liver cirrhosis. Applying logistical regression, we found VOC markers that correlated strongly with established clinical markers of liver function and also with limiting. These results are shown in the correlation matrix, where the size of the circles corresponds to the strength of the correlation and the color corresponds to the direction of the correlation for pairs of biomarkers. As these results are somewhat preliminary, we're waiting to reveal the names of the VOCs until we can confirm their identities with standards. We also identified the top 5 VOC classifiers that were also not collinear with one another, one of which was limiting, and constructed the ROC curve shown to the right. Compared to limiting alone, which I showed earlier in this presentation, this panel of 5 VOCs increased the area under the curve from 0.78 to 0.94. Sensitivity increased from 73% to about 90%, and specificity increased from 78% to about 92%, indicating a high diagnostic accuracy for discriminating liver cirrhosis. Hopefully, this case study has helped to illustrate the variety of ways in which you can tailor your search for non-invasive biomarkers of disease. For example, using the breath biopsy OMNI assay, one can perform non-targeted VOC biomarker discovery and targeted VOC analysis from a single set of samples. Upon validation of markers, a panel of VOCs could be developed for further screening. In addition, for markers that are not normally found at significant endogenous levels, an evoke probe strategy may be useful for non-invasive testing for disease, for monitoring disease progression, or for testing the effectiveness of therapeutic intervention. This second case study explores the role of on-breath VOCs in precision medicine for asthma. This example is broadly applicable to other chronic inflammatory airway diseases, where differentiation of subtypes can help better direct appropriate treatments to patients. Asthma is caused by activation of a wide range of inflammatory pathways that result in significant reduction in pulmonary function and quality of life. Globally, more than 300 million people suffer from asthma, with more than 25 million in the US alone, where it accounts for more than $50 billion in healthcare costs each year. Disease management is focused on achieving control of asthma symptoms to minimize the risk for future exacerbations. However, many patients do not respond sufficiently to treatment, and their symptoms are not adequately controlled. As a result, exacerbations often lead to visits to the emergency room or to hospitalizations, which further add to the burden of the disease. Diagnosis and deployment of effective treatment options is a major challenge, despite an increase in the number of available therapies in recent years. This is due to a lack of stratifying diagnostics that could better inform to which treatments the patient will likely respond, mainly because it is very challenging to directly sample the biological activity occurring in the lungs without very invasive, complex, or stressful approaches. This has effectively resulted in a trial and error type of approach to better symptom management. In addition, the treatment of asthma has been challenging because historically it has been approached as a single disease, with treatments applied universally to all patients based on their observed phenotype. However, asthma is now considered an umbrella diagnosis for a collection of heterogeneous diseases that manifest with similar symptoms but require different treatments. This collection reflects a complex biological network of distinct yet interrelating inflammatory pathways that can be used to differentiate subtypes of pulmonary inflammation. These inflammatory subtypes can be further divided into different endotypes, each defined by a distinct biological mechanism that links clinical phenotype to molecular pathways that can be targeted for treatments. Therefore, the precise definition of these endotypes is central to asthma management due to inherent therapeutic and prognostic implications. Breath analysis is being explored as a route to discover biomarkers to identify asthma endotypes, enabling a precision medicine approach to asthma treatment. And there is compelling evidence that breath VOCs can be used to classify patients by phenotype with high accuracy. In a moment, I'll briefly describe results from a large clinical study that was done by our academic partner at the University of Liège in Belgium. In this study, they focused on identifying exhaled breath VOCs that could classify asthma inflammatory subtypes, specifically eosinophilic and neutrophilic asthma. These two subtypes are defined by the gold standard measurement of sputum eosinophil and neutrophil proportions, and each broadly represents specific endotypes referred to as Th2 or non-Th2, depending on whether inflammation is driven by the subset of T cells. Eosinophilic asthma represents the Th2 endotype, and neutrophilic asthma represents the non-Th2 endotype. This classification is important as it will help inform which treatment the patient will receive. For example, Th2 asthma is generally responsive to inhaled corticosteroids, whereas non-Th2 asthma is not. The study demonstrated that 4 exhaled breath VOCs could distinguish between asthma inflammatory subtypes with reliability that was comparable to existing tests. In the upper right, we see that a small subset of VOCs showed good performance in discriminating between eosinophilic and neutrophilic airway inflammation in both the discovery and validation studies. In the lower right, we see that the small subset of VOCs can also discriminate eosinophilic asthma from other subtypes of asthma, including both neutrophilic and paucigranulocytic asthma, the latter of which has normal levels of both eosinophils and neutrophils in the sputum. The ROC curve summarizes the classification accuracy of these breath biomarkers alone or in combination with existing clinical markers. The purple line represents the VOCs, the orange line represents the blood eosinophils, and the light green line represents exhaled nitric oxide. All three sets of markers gave essentially the same performance in this population. However, if you combine the tests, we see a further improvement in performance, indicating that there's a lot of orthogonal information captured by these different sets of markers. It may also suggest that breath VOCs add to the phenotypic signal in these patients. Not shown on this slide is the ability of these breath biomarkers to discriminate neutrophilic asthma from the other asthma phenotypes, which performed well with an AUC of 0.73. This finding is of interest because there are currently no markers that can accurately classify the neutrophilic asthma subtype. In summary then, an analysis of exhaled breath from a large asthmatic population identified biomarkers that can accurately classify patients by disease subtype. And this has major implications for improved management of complex inflammatory diseases by enabling a better alignment of appropriate treatments to patients who are more likely to respond favorably. Owlstone Medical's respiratory panel. This is a research use only offering intended to support clinical research on chronic inflammatory airway diseases. The panel consists of a set of VOCs with clear relevance to inflammatory respiratory disease. These compounds can be easily collected on breath to enable a direct characterization of disease biology in the lungs by non-invasively sampling the airways. Components of the panel were selected from collaborative studies in the respiratory space and augmented through the exclusive in-licensing of additional biomarkers from the University of Liège. These are the same biomarkers that were just presented in a case study seeking to characterize asthma subtypes. Importantly, structural signatures for these molecules, based on authentic standards, are fully represented in our high-resolution accurate mass library. This ensures accurate identification of panel molecules and greater confidence in the results of the identified VOCs. And lastly, we offer the flexibility of analyzing for the respiratory panel VOCs alone as part of a focused service offering, or in conjunction with our discovery services using the breath biopsy OMNI assay. This panel will be of special interest to the biopharma community, where it can be used to support therapeutic development through characterization of disease endotypes, patient stratification, and monitoring of therapeutic response. While the panel currently includes at least 17 VOCs with mechanistic links to the biological processes, we intend to develop this as a dynamic panel. Thus, we will continually update the panel over time as scientific data are published and new information becomes available. This panel currently encompasses 5 general chemical classes and provides a readout of key biological processes that are frequently aberrant in respiratory disease, including oxidative stress, lipid peroxidation, inflammation, amino acid and fatty acid metabolism, and microbial activity. In this last case study, we'll look at how on-breath VOCs can be used to inform on malignant phenotypes in a high-risk population. Lung cancer is the leading cause of cancer death in the US and Canada. Early detection is critical for improving treatment options and survival. Survival rates are much greater when lung cancer is diagnosed and treated in the early stages before it spreads. However, because lung cancer doesn't cause obvious symptoms in earlier stages, diagnosis often comes only after it has spread, and survival rates plummet accordingly. Smoking is an established risk factor for lung cancer, with up to 50% of cases occurring in former smokers. Despite having stopped smoking, former smokers retain a significantly increased risk of lung cancer. Lung cancer screening can identify cancer at early stages and reduce lung cancer deaths. 2 large clinical trials have shown that low-dose CT scanning can reduce relative rates of lung cancer mortality by 20% or more. This is driven largely by a shift in the proportion of detected cancers from later stages to earlier stages. Despite the large reduction in deaths, low-dose CT scanning still isn't widely used because of significant costs and a high rate of false positives, where most lung cancer nodules are deemed to be benign. Moreover, eligibility is limited to high-risk populations, mainly older adults with significant smoking history. An accurate non-invasive breath test could be used in combination with current risk criteria as a pre-screening tool to determine eligibility for low-dose CT scanning. It also has the potential to expand CT scanning to lower risk populations, including those who have never smoked. And so, in conjunction with Stephen Lamb, Ronelle Myers, and other researchers from the British Columbia Cancer Research Institute in Vancouver, we set out to explore this possibility in a small feasibility study. I want to mention that Ronelle has presented this work as part of her keynote address at the 2020 Breath Biopsy Conference hosted by Owlstone Medical, and a recording is available through our website. Breath samples were collected from participants in the Pan-Canadian Lung Cancer Screening Trial. Subjects in our study were divided into 3 groups. The first group consisted of screen-detected cancers that were biopsy confirmed. The second group had indeterminate nodules. These are nodules that carry a moderate risk for malignancy based on the characteristics of the nodule and the individual's demographics. The third group consisted of participants with nodules that were stable for more than 5 years, at which point all nodule types are deemed benign and are no longer followed. Participants were 55 to 80 years of age, had smoked for about 35 years on average, and had quit smoking for about 15 years on average. Breath samples were analyzed by TD-GC-MS, though on a previous version of the platform, not the OMNI assay. In this study, 67 VOCs were identified, of which 22 were significantly different between case and control using a non-parametric 2-sample test, and 8 remained significant after accounting for multiple testing. However, because this was a small study, we utilized the unadjusted p-values to determine the level of significance. Among the VOCs that were different between individuals with confirmed cancer and controls were a group of 5 alkanes and 3 aldehydes. Each of the 3 aldehydes shown on the right in the box plot were elevated in the breath of subjects with lung cancer compared to controls, and levels in subjects with indeterminate nodules were intermediate between the two. The increased abundance of on-breath aldehydes in cancer patients observed here is in line with previously published reports. We speculate that the higher metabolic output of cancer cells drives an increase in reactive oxygen species production, resulting in an increase in oxidative stress and subsequent lipid peroxidation. As mentioned earlier, many of these lipid peroxidation products are volatile compounds that can be measured on breath. 5 alkanes shown on the left were significantly different in lung cancer compared to stable controls, with 2 of the 5 increasing and 3 decreasing with lung cancer. Like the aldehydes, the abundance of alkanes in subjects with indeterminate nodules were frequently distributed between the lung cancer cases and controls. Also, like the aldehydes, one mechanism that drives alkane production is oxidative stress, where the production of reactive oxygen species leads to the peroxidation of polyunsaturated fatty acids in membranes. At this time, it's difficult to assess the biological impact of the differential changes in direction of the alkanes in this study. However, the results may reflect differentially located changes in oxidative stress and lipid peroxidation, or perhaps changes related to specific patterns of unsaturated fatty acids. We looked at the classification potential of the VOCs identified in the study using different classification algorithms and a nested cross-validation approach. We determined that a random forest using 3 VOCs achieved the best classification accuracy. The final model was then refitted on the full data set using simple 5-fold cross-validation, and an average ROC curve was obtained. The resulting model attained an accuracy of 80% with a sensitivity of 75% and specificity of 80%. We're keenly aware that this is a pilot study that is limited by a small sample size, and this approach could be over or underestimating performance. However, the data are highly encouraging and demonstrate that value can be provided when incorporating breath biomarkers into lung cancer screening strategies. In conclusion, applying non-targeted profiling of breath VOCs to a population of former smokers identified several promising candidate biomarkers for lung cancer. The identity of these markers suggests that they are mechanistically linked to underlying oxidative stress and lipid peroxidation associated with enhanced metabolism of proliferating tumor cells. Of course, as with any study, these findings need to be validated. One recommendation is to increase the study power, and powered calculations based on the current data set show that a minimum of 40 to 50 subjects per group would be beneficial. Moreover, any follow-on study that utilizes Owlstone Medical's OMNI assay will benefit from the high-resolution accurate mass platform and a growing spectral library of standards. This study also demonstrated the feasibility of incorporating breath biomarkers into lung cancer screening strategies, for example, to identify high-risk populations. Modeling data from this study suggests that just 3 markers provide very good accuracy in discriminating cancer from controls, and other data showed that subjects with indeterminate nodules had levels of breath biomarkers that were intermediate to the case and controls. The value of breath VOCs to predict cancer risk could be further assessed by following the indeterminate nodule group over time to identify those that develop cancer or are confirmed as benign. Finally, given the ease of non-invasive sample collection and the promising accuracy indicated by this feasibility study, breath biopsy may reflect a new modality for pre-screening at-risk individuals and to help manage their clinical care. We hope this webinar was informative and provided a clear understanding of Owlstone Medical's breath biopsy OMNI assay, which provides the broadest analysis of compounds on breath. The OMNI assay is part of our breath biopsy services that can support every aspect of your breath biomarker research, including clinical trial design, breath sample analysis, and expert biological interpretation, along with biomarker validation and test development. The centerpiece of our OMNI assay is a high-resolution accurate mass TD-GC-MS platform. This provides a high dynamic range that enables detection of both high abundance VOCs and trace level analytes to maximize the number of compounds measured in your breath samples. Biomarker identification uses our breath biopsy library of authentic standards to provide high confidence molecular identification. Incorporated at all stages of the project is rigorous quality control to ensure you get the best possible results and to maximize your project's success. For researchers with a specific focus on chronic inflammatory airway disease, we also offer the focused breath biopsy panel for respiratory disease, which can be run as a standalone service or in combination with the OMNI assay. I provided several examples today of how breath biopsy can enable biomarker discovery for early disease detection and precision medicine. While I focused on diseases of the liver and lungs, this approach has application to a broad range of cancer, inflammatory, and infectious disease areas, some of which are listed in the condensed table to the right. This table represents a cross-section from a review of more than 100 peer-reviewed publications using Owlstone medical products in biomedical research applications. And a full list of publications is available through the resources link on our website. Should you have any questions about the content of today's presentation or wish to speak with us regarding how breath biopsy may be incorporated into your clinical research or trials, please contact our business development team using the email addresses shown here or by placing an inquiry through our website. Additional information related to the science and applications of breath biopsy are outlined in our ebook, which is also available for download through our website. I also would like to mention that we have a number of opportunities available to contribute to the development of technology and applications of breath biopsy in the biomedical space, both here in the States and in our Cambridge, UK headquarters. Further information on each position can be found on our website and LinkedIn page. On behalf of our entire organization, I want to thank you for your attention throughout this webinar. We look forward to speaking with you very soon.
Thank you very much, Jeff and Robert, for this presentation. I'd now like to open the floor to questions. So please do remember to send us your questions using that feature of the platform. So before we do start with the first question, I did see someone ask whether it would be possible to get the references for the slides that show different disease types. I can confirm that will be possible, so we can send you across some references afterwards. So the first question we have, and I believe this is a question for you, Robert, is the issue in NASH drug development is the identification of prognostic markers, so progress from F1-2 to higher grades of fibrosis. How would identification of VOCs support the discovery of prognostic markers, also given the presence of concomitant metabolic diseases in NASH?
Yep, I'll try to address that question. So in the presentation we gave today, we talked essentially about the bookends. Can you differentiate cirrhosis from healthy controls? Our next step is to take that and kind of work our way back. So start looking at disease progression there. So especially with the evoke probes, where we think we get a better signal compared to just endogenous molecules by themselves, we would like to get a much larger clinical trial going of individuals with individuals at each of the different stages of progression. Of course, ultimately, I think we will end up using multiple probes for that kind of test. So something like limiting by itself may or may not differentiate the different stages of fibrosis or liver disease progression, but those could be supplemented with either other endogenous molecules or perhaps even other evoke probes against specific targets that become important during those different stages of progression.
Great, thank you. And another question that came through during your talk. So is there evidence for quantitative differential P450 metabolism of an evoke relative to age, concomitant diseases, etc.?
So we haven't yet tested that. We're in the process, so we're just starting into some of the human clinical trials with our evoke probe there. So we don't have that information, but I would guess based off of existing information in the literature that there will be some influence of those parameters on the P450s that were in question there.
Thank you so much. And I believe, Jeff, this question is for you, and if you're able to turn on your webcam, which is it possible to use the device at home?
So right now it does need to be used in a clinical setting. It really is designed for discovery, but we do have other technologies that may be more amenable to a point of care testing that get further along in development.
Thank you. And the next question is, does Owlstone have any regulatory approvals, so IVD or PMA, for its breath biomarkers? If not, does Owlstone see its system being used for IVDs, or do you prefer to develop it as research use only?
Yeah, I can take that one. Currently, we are using it as research use only. Our ultimate goal is to develop a clinical test, and so we would want to get that as a IVD and through regulatory approval.
Great, thank you. And next question, if someone, so alkenes have been a mainstay of retention time indices in GCMS for decades. If alkanes and alkenes have biological importance, what are you using to standardize retention time?
That's a question that I probably don't have enough insight in, but you could easily use labeled standards for the retention time indices.
Great, thank you. And two sort of similar questions, so I'll ask them together. So the first is, how does diet influence the concentration of markers? And then someone has also asked, oh, actually, no, sorry, the other question that person has left. So how can diet be related to the concentration of the markers?
Sure, so I'll take that one. So certainly diet will have an impact on some of the markers. And it's just like any other clinical test that's being done, whether it's measurement of biomolecules in plasma, urine, or other biofluids. Now, we're not aware of any really high quality, rigorous studies that have been done to look at various diets on the different markers, but we can anticipate there would be some. We also have knowledge of what some of these markers are and where they're derived from, and so we do have that knowledge going into some of these studies. Ultimately, though, I think concern about diet would only be a problem if you would expect there to be systematic differences in diet between the groups of interest. So if the healthy controls were on one type of diet and a disease population was on a different diet, then that would clearly have some impact on the markers you would detect. And I think the last point to make on that is that we do collect information related to the time since last eating as part of the CRF.
Great, thank you so much, and thanks everyone for your questions. We've got a few more coming through, so do please continue sending them across. The next one, if either Robert or Jeff wants to comment, is for liver disease. Has a biomarker that correlates with changes in ALT been detected? This would pull in detecting drug-induced liver disease.
I'll try that one. I don't know if if we have found one or not. We certainly have the data to answer that question, but off the top of my head, I don't know. That ALT, based off the data we found, or based off the data we have, we weren't that interested in ALT, so we haven't really explored that to in a lot of detail that I'm aware of.
Great, thank you. The next question, so hello and thank you for very interesting presentations. I have a question regarding asthma inflammation endotypes identification. We know very well that most biomarkers are very unstable through the time. Do you have any data regarding the stability of VOC in asthma patients? Thank you.
We do not have data that I'm aware of it's for stability in asthma patients, but we have collected other data in general terms on stability. And we feel that the processes we have in place are particularly for the time courses we're looking at.
Great, thank you. Next question, is the respiratory panel only directed towards airways disease, or has the panel been evaluated in fibrotic lung diseases also?
The respiratory panel is really meant for any kind of inflammatory type lung disease. So you potentially could look at that as well. Keep in mind that this is essentially a panel that's been pulled together from the literature as well as some of the biomarkers from the University of Liège study. And so it's really intended to provide a readout of general inflammatory processes in respiratory disease.
Thank you. If a sample is thought to be contaminated or if contaminated with exogenous VOCs, is it possible to extract and clean the data to remove these?
Very likely not. I guess you could, if you had if you knew what exactly the background levels were, if you had taken a background sample that had the contaminants in it, potentially you could do a subtraction of the data, but theoretical versus practical are two things. So I'm not sure on that one.
And can you collect long non-coding RNAs?
Yeah, so our focus has been only on, well, primarily on the volatile organic compound portion of breath. We talked a little bit at the beginning of, I think, my introduction, maybe even Jeff's, about the other fraction of breath where we have the potential to collect things like DNA and RNA and proteins and so forth. We started looking at that. Yes, you can detect RNA and DNA. The problem is, though, that you would probably require a lot more breath to do so. The amount of RNA and DNA that we were collecting compared to things, if you were just to compare it to like PCR-based methods to look for presence of pathogens, for example, we were very low on that curve. So we have further work base.
Great, and we've got a couple more minutes. We're still getting lots of questions through, so I hope you don't mind. We'll just ask a few more. So the next is, how long can you store the desorption tubes after collection, and how many times can you safely recondition them and still have reliable results?
That's a great question. I don't know if I have the technical details to answer that. Yeah, so I have to pass on that. We actually recondition our tubes a number of times, but we do keep track of the number of reconditions that take place for each tube. Since they're barcoded tubes, so we track the entire history of that tube.
Thank you. And how do the VOC profiles between pre and post removal of lung tumors compare in the same patient? Is there anything you can comment on there?
Yeah, I don't have that information. I don't think we've collected that information, so I really can't comment.
Thank you. Is there a washout effect for VOCs present in only low concentration with repeated breath sampling?
Can you repeat that again?
Yeah, of course. So is there a washout effect for VOCs present in only low concentration with repeated breath sampling?
Great question. If they're endogenous molecules and they're low level, I would expect they would retain their low levels and not necessarily be washed out or dropped with repeated sampling. But I haven't really seen data on that, so it's hard to answer.
No problem. And you mentioned the additional value to VOCs and FeNO in asthma. Could the analyzer assess both from the same sample?
VOCs and FeNO, is that what the question was?
Yeah, that's right.
Yes, you should be able to. Well,
Great.
you wouldn't get it from our sample, but you could get it from the same patient collected at the same clinical visit.
Perfect, thank you. And we're running close to time now, but if you don't mind, we'll ask just one or two more questions. And then if any other questions do come through, I will send them across in an email afterwards, and I'm sure Owlstone will be happy to get back to you. But the next question was, do you need to genotype CYPs for the limonene assay?
Good question. I think that's something that is of interest to us. Yeah, I would say it'd be good information to have.
And we will have the last question being, is it intra-individual or intra-individual or inter-individual variability for these VOC biomarkers? Sorry, a bit of a tongue twister there.
So I think the question is, repeat again, I think the question is primarily asking which is greater, the inter-individual. Cross individuals are within an individual sample over time. Is that possibly the question?
I think so. They've followed up to say yes. So I think that's okay. The right understanding.
Yeah, like other clinical measurements of biochemicals, we would expect that variation across individuals would be greater. So, for example, a lot of metabolomic studies using plasma metabolomics, typically you could see anywhere from 35 to 50% biological variation across individuals. The variation within an individual would primarily be driven by things like diet over time, exposure to the environment, et cetera, over time, changes in circadian patterns and so forth. But individuals should be more closely related to themselves than they would be to other individuals in terms of their VOC patterns.
Perfect, thank you. So I think with that, we'll bring the session to a close. The last comment through from just someone is excellent presentations, great and exciting work, and they'll look into the approach in more depth. So thank you so much for the very informative presentations. If anyone does send through any last questions whilst we're concluding, as said, I'm sure Owlstone will be happy to get back to you after the webinar's concluded. So thank you once again for your questions, and thank you once again to Jeff and Robert for the excellent presentations. Owlstone Medical are also participating at our Biomarkers Congress in November, so if you'd like further information on that, please do get in touch and visit the conference website. And on behalf of Oxford Global, I'd really just like to thank everybody for joining us today. And I'd like to say a big thank you once again to the team at Owlstone Medical, who put a lot of time into making sure the webinar was as interesting and valuable as possible. So that all remains to be said is have a great day, and we really look forward to welcoming you to one of our future events. Thank you all.